Hello. Today we'll start our seventh topic of multisystem 1, and we will talk about the properties of antibodies. In this topic, we'll talk about, we'll give briefly the development of the B lymphocytes, which actually the main precursors, and which upon differentiation will give plasma cells, which will secrete the different types of antibodies. And then we'll talk about the structure and the function of each type of the antibody, G, M, A, E, and D. For the B lymphocyte, the B lymphocyte starts development in the bone marrow, and then it needs to migrate to secondary lymphoid tissue. So you can see from the figure, it can migrate through the circulation into different secondary lymphoid tissue, either lymph node, pairs, patches, or even the spleen. So basically, there are six functionally distinct phases in the development of the B lymphocyte. The phase one, there are three phases in the bone marrow, and there are three phases in the secondary lymphoid tissue. In phase one, there is like a repertoire assembly in which there is a generation of diverse and clonally expressed B cell receptors in the bone marrow. So you have B cells that express different receptors, and we'll soon understand after that how the different stages of the B cells are identified by the assembly or the repertoire assembly of these expressed receptor on the surface of the B cells. Then you have phase two, in which there is an alteration and elimination of the inactive B cell receptor that can bind to the component of the human body. In other words, there is a negative selection toward B cells that can identify self-antigen, because it's crucial to have B cells that can differentiate between self and non-self. So the negative selection is important phase in the bone marrow to eliminate the self-reactant B cells to our own human body molecules. Then phase three, in which after the negative selection, in which there is a positive selection and the promotion of the fraction of these immature B cells to become mature, and then it will be enabled to leave into the circulation to migrate to the secondary lymphoid organs. After that, phase 4, which starts in the secondary lymphoid tissues, in which it searches for the infection. So there is a recirculation of the mature B cells between lymph blood and secondary lymphoid tissue, patrolling to search for infection. Once the infection is found, there is an activation and the clonal expansion of B cells. This actually is done by the pathogen-derived antigen in the secondary lymphoid tissue. And it's crucial for this activation to fight the antigen. And why is clonal expansion? Because each B cell actually divide and expand and proliferate from a single clone. And keep in mind, every single clone of B cell will generate a specific type of an antibody for a specific antigen. For phase 6, this actually, once it found the antigen, found the infection, it will attack this infection. So these B cells will differentiate to the famous plasma cell, which is the antibody secreting cells, and will give as well memory B cells in the secondary lymphoid tissue. And these are the 6 functionally distinct phases in the development of the B cells. So we can see here, in the course of the development of B cell in the bone marrow, different stages or different forms of the B cell precursor, starting from the pluripotent hematopoietic stem cells that give rise to common lymphoid progenitor, then the B cell precursor, then the pro B cells. Actually, you can say that it can be differentiated or identified by the different combination of the CD proteins on its surface. So you can see each type of these B cell is differentiated by the repertoire of the receptors, or the collection of the receptors on its surface, or the surface protein of CDs. You can see that pro B cells is very have a well identified CD 127, and actually it is the alpha chain of very important cytokine, which is the interleukin 7, and we'll understand later why there is a specific receptor for interleukin 7 on the surface of the pro B cells. So the early stages of these B cell development actually are dependent on the bone marrow and the stromal cells. And the interaction of these B cells with the bone marrow and the stromal cells by integrins and adhesion molecules facilitates and directs as well its proliferation. So you can see here the different stages of the B cells, starting from the lymphoid progenitor cells until reaching the late, the early, then late, then pre-B cells, are differentiated, as we said earlier, by the the type of the, or the kind of the receptor on its surface. And it is still attached over all these stages, it is still attached to the bone marrow stromal cells. And this is crucial to have these cells attached on the bone marrow stromal cells to make sure, before releasing these B cells to the circulation, to be sure that it can differentiate between self and non-self. Because in case that these B cells are released in the circulation before the negative selection and the positive selection, the B cells will be able to identify, if it's released before these selections, the B cells will be able to identify self molecules of human body, and this may lead to autoimmune disease. In the near later stage of the development, at the pre-B cells, the stromal cells starts to secrete the important interleukin, which is interleukin 7, as you can see from the figure. This interleukin 7 will help to release the pre-B cells from the attachment from the stromal cells into is still inside the bone marrow, and now it's termed immature B cells, and you can now it is expressed one of the types of the antibody, which is immunoglobulin M. So one of the markers of the immature B cell stage in the bone marrow is the expression of the IgM on its surface. So you can see that, as we said, at a later stage of stimulation, B cell requires interleukin 7 to stimulate their proliferation. But keep in mind, it is still at this stage, it is immature, and it needs to be selected towards the non-self recognition. So you can see here, initially, in case these are cells which are immature B cells in the bone marrow, in this case, you can see that this type of cells, there is no self-reaction, there is no reaction with self-antigen. And so in this case, if the bone marrow is sure, and in the mechanism in the bone marrow, it's sure that these immature B cells is non-reactive to self-antigen, these immature B cells will be released into the blood, and will express two types of antibodies on its surface, IgM and IgD. So again, these immature B cells that are not specific for self-antigen in the bone marrow, they are further mature and further express IgM and IgD and leave the bone marrow. While in cases that the immature B cells are specific, for self molecules, and this is being actually done and aided by the bone marrow stromal cells. So generally these cells express a surface marker that in case these immature B cells recognize this surface marker, so the bone marrow understand that this clone of B cells can recognize self. So in this case, these immature B cells are retained in the bone marrow and is not allowed at all, this B cell, to leave into the circulation. So here you can see that, again, if the immature B cells specific for monovalent self-antigen, they develop a state called energy, okay? What's energy? Actually, the energy, it is the lack of reaction by the body defenses mechanism to foreign substance. So actually, it's a kind of a mechanism of protection. So this type of cell will go into energy state to avoid the reaction with self. These energetic B cells have actually shorter life time than the normal B cells, from 1 to 5 days versus the 40 days half-life time for other functional B cells. So this tolerance can be checked, or this central tolerance can be checked within the bone marrow. So it will, and this will lead to the removal of the anti-self or these energetic B cells. So it's a kind of a screening inside the bone marrow to make sure that the functional B cells are only released into the circulation. While the peripheral, so you have a central tolerance which are employed inside the bone marrow, where you have another tolerance. It's a kind of like another layer of double checking, which is called peripheral tolerance, in which, in case that the anti-self B cells have been leaked out the bone marrow, so there is another mechanism which is called clonal deletion, which actually apoptoses, leading to the death or the programmed cell death of these energetic B cells, or these cells that can recognize self-antigen. So you can see here that B cells is signaled, as we said, it can express IgD as well, because initially it is to express only IgM. So B cells can be signaled to make IgD and to become unresponsive to antigen. And this process is called class switching. And we'll understand more in the immunogenetic lecture how this class switching occur. So basically, what's meaning of class switching? It's basically the ability of the B cells, instead of synthesizing and secreting a specific type of antibody, it will be able to rearrange its gene sequence, gene segments, to generate another type of the antibody. In this case, it's IgD. So in this case, the generation of the IgD will help these B cells to be energetic B cells, and it is either have a central tolerance or peripheral tolerance. So it will go into the peripheral circulation, but as we said, it will not survive for long. So let's see here the routing of these B cells, the immature B cells, once these B cells are being released from the bone marrow. They are still immature, and now and for sure they can differentiate between the self and non-self. So you can see the B cells. Here is the example as a lymph node. you can see the B cells enters into the lymph node, generally through the blood vessels, and from the high endothelial venule, it can reach the T cell area, and then it will move to the B cell area, of course, upon the finding of the antigen, and we'll talk in details in the coming lecture how this occurs, and even in the coming slides. So you can see its route is being activated by the exposure to the antigen expressed and activated by the T cells as well. And if they don't encounter any specific antigen, they will leave the lymph node and exit through the efferent lymphatic vessel. Because the antigen can be entered to the lymph node through the afferent lymphatic vessel or even through the blood. But the main point here that the routing of the B cells through the secondary lymphoid organ will determine if these B cells will mature and differentiate to plasma cells to secrete antibodies. And the main cause here is the exposure to the antigen. So in case there is no exposure to antigen and there is no specific antigen, these B cells will leave the lymphoid and will keep on circulation in the lymphatic vessels and blood. So here we can see how it's got activated once it is it reaches the secondary lymphoid organ. So you can see here the yellow cells here, it is the B cells. So you can see it's moving into the capillaries of the secondary lymphoid organ, and upon the secretion of the chemokine CCL21, this will attract the B cells to this region of high endothelial venules. The release of the another important cytokines, which is the CCL19, together with the CCL21, will attract these B lymphocytes outside of the venule toward the lymph node. And so you can see now that the B cells have escaped from the venules into the tissue of the lymphoid organ. Another cytokine common action, which is the CXCL13, it will attract these B cells into the primary follicles. And actually the need for the attraction in the primary follicle actually to get the chance if there is any antigen to be sensed and hence to be activated these B cells. So you can see here the interaction between the follicular dendritic cells and the cytokines in the presence of the antigen will drive the maturation of the immature B cells. And we can identify the mature B cells by starting the expression of the IgM and the IgD on its surface. And now these B cells is mature. And in this case, these mature B cells will leave the lymph node and it can recirculate between lymph, blood, secondary lymphoid organ. And soon after that, it will be differentiated into plasma cells and start to secrete specific antibodies toward the specific antigen. And if these mature B cells, as I said before, do not encounter their specific antigen in the follicle, they leave the lymph node and continue to recirculate through the secondary lymphoid tissue via lymph and blood. And these immature B cells that fail to enter the follicle continue recirculation as well, but obviously it will die because it will never encounter an antigen. So actually, as we said, this activation of B cells actually is antigen mediated, and the differentiation occur in secondary lymphoid tissue. So you can see here, as again, the internalization through the blood vessel of the B cells. It's entered, as we said before, and these B cells once it matures, it encounters antigen, which actually delivered to the lymph node through the afferent lymph draining from the infected tissue. The B cells as well need activation by T helper cells to form a primary focus of the dividing cells. So you can see these cells soon migrate upon activation by the T helper cells. You can see initially the B cells came out as a result of the cytokines we said in the previous lecture, in the previous slide. So you can see it is recruited from the HEV, and then it is activated by the T helper cells. It is exposed to the antigen here in the T cell area. Soon after that, it is start to divide. It move to the B cell area where it form germinal centers with a lot of the B cells dividing and start differentiating. Once it is differentiated into the plasma cell, which have the capacity to secrete the specific antibody for the antigen, it will soon, as you can see, the plasma cell, it will soon migrate back through the circulation to be able to secrete these specific antibody into the circulation. So this activation leads to the activation, by, of course, by the T helper cells and the exposure to the antigen, leads to the proliferation and differentiation of these mature B cells into plasma cells. So, and leads to the migration, as we said from the figure, it leads to the migration to a primary follicle, as you can see, then to move to form a germinal center where they further divide and differentiate. The B cells from this germinal center migrate either to the medullary cords or the bone marrow to complete their maturation into plasma cells. So we can see here, that's like a simplified figure. You can see to see how this differentiation occur. Initially, you have different clones of B cells, and the invading antigen binds to the antibody which is expressed on the surface of the B cells. You can identify the different clones of the B cells by the color of the antibody attached on the surface. So the invading antigen actually selects which clone of the B cells will be amplified and undergo proliferation. So this invading antigen binds to the antibody on the surface of just one clone, specifically of just one clone of a B cells. Soon after that, these B cells, this clone of the B cells, will be selected by this antigen, and it will start, as you can see, it will start to form a large clone of genetically identical B cells are produced. So you can see here the expansion of the number of the B cells that is able to express on its surface a specific type of antibody toward this specific antigen. Soon after that, you can see that these B cells is differentiated into two types of cells. A plasma secreting cells of the same antibody which was originally expressed on the B cells, and the memory B cells which will confer immunity over a long term and will be able to secrete antibody again in a shorter period of time toward the same antigen. These plasma cells, of course, will release antibodies into the blood. So you can see here that any abnormality will lead to different types of B cell tumors, okay? And these different B cell tumors reflect this heterogeneity of developmental and differentiation states in normal B cell population. So you can see that from the table, each type of tumor actually correspond to a normal state of B cell development. So you can, in other words, we have different stages in the differentiation of B cells, either late pre, early pre, you have immature forms, you have the mature forms. Any abnormality can lead to a specific type of tumor related to this stage. So tumor cells have actually similar properties to the normal cell equivalent. And they migrate, actually they behave the same. They migrate to the same site in the lymphoid tissue. And they have actually as well similar pattern of expression on each surface of the glycoproteins, maybe more and highly expressed same pattern of these cell surface glycoproteins. So why these occur? Because actually the tumor can arise from mistakes in the gene segment rearrangement, and this can lead to activation of some of the proto-oncogenes to be an oncogenes, and they found that these types of oncogene will affect the rate of cell growth, the rate of division, and the rate of differentiation, and will eventually lead to progressive proliferation of the peculiar type of the cell, leading to cancer. So as we already said, actually B cell tumors are actually clonal, so it comes from one type of cell. So generally tumors that are derived from naive B cells. What's meaning of naive B cells? That means B cells that is not still not encountered by the antigen. So any tumors derived from these naive B cells will grow in the follicles of lymph node and will develop types of lymphomas, while the tumors which are derived from the plasma cells generally will develop myelomas. So you can see here from this table the different type, the name of the tumor, and the equivalent, the normal cell equivalent stage of these B cells, the location actually, where is the tumor, this type of tumor. Actually, it is based on the type of the B cell or the equivalent form and the status of the Ig variable genes. So you can see some of the tumor are affected by mutation of a region or a segment in the of the variable region of the variable the variable region of the genes that are responsible for formation of the variable region of the antibody. So so again, this table describes how the different types of tumor related to the different types of the cell, normal B cell developmental stages, as well as the location of each of these abnormality, and as well as the status of the immunoglobulin variable genes.
Doctor, do we have to memorize each type?
Now we'll talk about the structure and function of the 5 different types of the immunoglobulins or the antibodies. So starting with immunoglobulin G, actually it's composed as well as typically from L chains and the heavy chains, and actually the total molecular weight of this IgG is around 150,000 Dalton. There are 4 subclasses exist either from gamma 1 to gamma 4, and these actually are differentiated by slight changes in the amino acid sequence on the H chain, on the heavy chain. Its function include, actually, before we say its function, it's the IgG, actually it is the highest serum concentration of all the immunoglobulins, and it can adhere to cells that have the receptor for the Fc region of the IgG, and it can fix its complement, if you remember, in the process of opsonization that result in a cell lysis. As well, IgG can mediate the placental passage of maternal antibody to the fetus. For the structure of IgG, you can see that the IgG have the ability to be cleaved in 2 regions by the effect of 2 enzymes, the papain and the pepsin. So you can see the papain can cleave this antibody into, actually you can see into 3 different pieces. So it will give actually 2 antigen binding Fab fragments. So you will have it can bind these Fab fragments as usual. It will bind to the epitope of the antigen, but it will it cannot enter into a lattice formation. It cannot precipitate or agglutinate the antigen, because it have two separate Fab regions. The FC fragment, you can see that upon the papain cleavage, it will have the disulfide bond, the marked by the disulfide bond. It is still can be able to activate the complement system, and they can as well control the catabolism of the IgG. It can fixes the IgG to the cells. And obviously, because it's the FC, and normally if the cell is attached to the antibody, the receptor on the cell will be the receptor for the FC region of the antibody. So it can be fixed, this FC region alone can be fixed on the cells. And as well, it can mediate placental transfer of the antibody. If the pepsin cleaves the IgG, it will give a totally different scenario. There is a totally different scenario in which the two Fab region will be connected by the sulfide bond, the disulfide bond, and the FC region will, all of the FC region will be degraded. And these two joined Fab fragments will be differentiated in the nomenclature, being said that Fab AB prime 2 to be able to differentiate it between the normal Fab region. And this actually is capable of lattice formation and removal of antigen because of the existence of the two Fab regions. And where the FC region is removed rapidly from circulation, then the intact IgG, as we said, the FC fragment is extensively degraded. So now we'll talk about the IgM. Actually, IgM exists in two structural forms: a monomer, which is actually synthesized by B cells and is retained, as we said before, on or expressed on the B cell membrane, and serves as a B cell receptor specific to and single specific epitope. And this, the hypervariable region of this monomer, differ for each B cell clone, because generally all the B cell clone initially will express IgM, but you need to understand the hypervariable region is different. Okay, the other form is the secreted form of IgM, and actually it exists as a pentamer, and of course it is secreted following the activation by the antigen and the cytokine activation of B cells and the differentiation of the B cells into plasma cells. So it starts secreting this IgM that can exist as a pentamer, and the hypervariable region of this pentamer are the same as those on the antibody monomeric receptor over the B cells. So since it's a pentamer, so theoretically it will have 10 possible epitope binding site on the pentamer. It has been found that 5 of these paratopes of high affinity, while 5 are of low affinity. And you can see here the structure shape of the IgM. So you can see additionally IgM has 4 constant regions instead of the 3 constant regions we have seen in the general structure of the antibody in last lecture. And it actually have the highest molecular weight, and it reached 1 million. And you can see that all the 5 molecules of the IgM molecule are linked together by a joining chain called a G chain. And the major functional properties is the ability, this IgM actually it is one of the it is the first, the earliest antibody to appear after antigenic stimulus, and works very much in fixing complement. What about IgA? So actually, it is a very peculiar type of antibody together with IgM, because it can exist as a monomer, as a dimer, and as a dimer plus secretory piece or secretory component. So you can see this figure shows the dimer, structure. It's not showing, it's only showing the dimer, it's not showing the dimer with the secretory phase. So this dimer actually can be transported across the respiratory and the intestinal mucosal barriers by the help of this SP region or a secretory piece, which is a receptor for the IgA FC region on the mucosal epithelium. So actually this secretory piece will exist in the antibodies that need the transportation through the respiratory and intestinal mucosal barrier. As well, this secretory piece, it protects the IgA from proteolysis, and there are two subclasses because each IgA, so it have alpha 1 and alpha 2 subclasses. While the functional properties, it's obvious, it will, this kind of antibody, it will found in a high concentration in secretions. It can, as we said, exist as a dimer with a half-life time of 5 days, and it protects the mucosal tissue, saliva. So you can see it's in all secretions, almost saliva, tears, even colostrum, by blocking, of course, bacteria and a lot of pathogen that can be that can affect our body. So in this figure, you can see actually how the secretory piece can be integrated into the structure of the dimer. So you can see here how the dimer form plus the SP domain is integrated to form the third structure figure of the structure organization of the IgA. And as we said, generally the J chain is responsible to, beside attachment of the two monomers, it's responsible for the polymerization and facilitate the polymerization of IgA and IgM, because as well J chain is present in IgM, while the secretory piece is very peculiar to the IgA, and beside its function said in the previous slide, it is responsible for the transportation through the mucous membrane of epithelial cells, because as we have just said, the importance of IgA in combating pathogen in secretions from our body, like tears and saliva. So let's talk about the structure properties of IgE. IgE as well has, as you can see, highlighted the region, the additional constant domain, and actually the carbohydrate content of it's around 18%, and this will increase its molecular weight to reach almost 200,000 Dalton. Actually, the IgA is very peculiar to be unstable at a degree around 56 degrees centigrade. And it's called regain because this antibody is very sensitive and involved in allergic reaction, and it will cause the release of histamine when it attaches to the antigen in tissue. Interleukin-4 is very important in mediating the class switch to IgE production. So that's mean this B cell originally is secreting or forming a specific other type of antibody. Upon exposure to interleukin-4, it will do class switching to start secreting IgE. This type of antibody is extremely low, have low serum concentration, and the half-life of its and half-life as well, because its FC region can bind avidly to tissue-bound mast cells and the circulating basophils as well via a specific receptor for this type of antibody, which is the FC epsilon receptor on these cells. And the binding of this antigen to these IgE sensitized cells, either basophils or mast cells, triggers the release, of course, of majorly vasoactive amines like histamines. And this will result in atopic disease disease characterized by hives, local reaction at the region, or anaphylaxis like the systemic reaction. Of course, this type of antibody doesn't cross the placenta or fix complement by the conventional pathways. And the binding of IgE to the eosinophils activated by interleukin-5 result into the major function of this type of antibody is the elimination of parasitic helminths. For the IgD, actually, it have a different structure classes, either delta kappa 2 or delta lambda 2, based on the isotypes. Its functional properties, actually, it's found on majorly on around the B cell membrane of 15% of the newborns and on the adult peripheral blood lymphocytes in conjugation of the IgM. If you remember, we have seen the IgM and IgD originally at the beginning of this lecture. Its serum levels are very low, half-life 2 to 3 days. The IgD receptor is on a B cell membrane, as we said earlier in this, as we have seen earlier in this lecture for antigen. Okay. For this table, summarize all the structural properties of the human immunoglobulins. You can see the different heavy chain isotypes. As we said earlier, it's either gamma, either mu, alpha, epsilon, or delta. You can see the different classes as well for the heavy chain for each antibody. You can see the different isotypes for the light chain as well for the different measure. It's either kappa or lambda, and as well you can see only the IgM and IgA that could be associated with the J chain. As we said, for the IgA there is a J chain and the SP region. You can see that the percentage of carbohydrates, and majorly IgE and IgD, have the highest percentage of the carbohydrates. As well, the molecular weight, and you can see it differs according to the presence, the structure of the antibody, if it's a dimer or a pentamer, or even with the SP region. You can see generally the largest molecular weight occur in the pentameric structure of the IgM. Here you can see the functional properties of the immunoglobulins, and you can see as well the average serum concentration of the different classes and the different types of the antibody, and you can see the major concentration of the major type that have the highest concentration is actually IgG isotype alpha gamma 1. The IgG gamma 1, while the lowest actually it's IgD in the serum concentration, the lowest, sorry, it's IgE. And as well, you can see the half-life time of each class of the antibodies, and as well IgE and IgD is the lowest half-life time. You can see that actually the antibody which is involved in the activation of the complement, it's only classes of IgG together with IgM, but majorly it's IgM. The other types, A, E, and D, is not involved in the complement activation. And as well, the ability to bind to the Fc receptor on the surface of the cells is limited to certain classes of antibody, excluding the IgA and IgD. The ability of the antibody to cross placenta by itself is generally a behavior that is related to the IgG antibody. So it's important to identify the difference between affinity and avidity. So affinity actually, as we all know, it represents actually the binding strength between one paratope and one specific epitope, while the avidity actually it's combined strength of all these binding sites on a single antibody molecule. So actually avidity can be considered as it is a collection of all the affinities of the antibody, as you can see from the figure. So here in the figure, this figure, you can see is the difference between affinity versus avidity. You can see here that there is a type, the IgG antibody can bind with affinity, with high affinity toward the epitope on a specific antigen. And here you can see the, although the pentamer, the IgM pentamer can bind with low affinity on each binding paratope, but if you calculate the avidity of all the molecule, it will be higher than the IgG. So generally in this example, you can understand that even if the affinity of the antibody IgG towards its antibody, towards its antigen is very high, but the avidity of the IgM with its pentamer structure is a lot more than the avidity of the IgG, because the avidity of the IgM actually it is a reflection of all the different affinities, which even low, but when you calculate it in total, the avidity will be more. This figure will describe the cross-reactivity, which is the ability of the same antibody and can bind or react to two different antigens, but of course the affinity will be different. So you need to understand that upon the exposure of an antigen, the first antibody is detected in the serum is IgM, which soon decreases with the emergence of the IgG antibodies, which can last longer, and actually it will provide like a memory for this type of antigen. Let's talk now about the antibody response to SARS COVID-2 in patient. So this line chart represent as you can see from the figure, represent the, hold on please, the SARS-CoV-2 RNA and antigen. So you can see here, this one, the blue line, hold on, represent actually the status of the virus inside your inside the patient, and you can see where is the onset of symptoms start to appear, on what level of the antigen of the virus, and as well when it starts to decline, and this actually is detected by PCR. So you can see there is a window period, you can see there is a start to decline here, and then it starts to recovery. In this scenario, you can see that the IgM, it starts to be detectable after first week of being exposure to the virus, and start to peak with peaking with the virus antigen in the serum, and as well it starts to decline until disappearance with the decline of the antigen of the source inside the blood. With the decline of the IgM, you can see that, as we said in the last slide, it will start the emergence of the IgG antibodies, and it will remain, as we said, in the blood and provide a long-term immunity. So you can see over days here the profile and the antibody response of IgM and IgG on the onset of the symptoms as well, and the antigen of the SARS. Together with this figure, which explains more what is the region that can be considered to be negative or positive. So if you have a blood test, the likely negative, and you can detect the virus in starting up till week 1 after have been detected by the PCR, while the likely positive for the corona test for the antibodies, of course you will not see negative antibodies, because you can see up till like almost the initial of the week, the last of the week 1, the antibodies didn't start to kick on. And after that, starting from the week, almost like the end of week 1, you can see the result of the antibody test will give rise to positive results, either IgM, IgG. And you need to understand for this result, the positive result of either IgG or IgM together or each of them alone, it will determine the status of the infection. So if you do a PCR, you can see the region likely to be negative or positive or likely negative. You can see that although here, because this is the detection threshold for the PCR, the solid dark line, black line, although it cannot be detected by PCR, but still there is a viral load that is below the threshold, but it's likely to be negative soon with the existence of the majorly the IgG antibody. And here you can see the slide for the test for the antibody. You can see that it is having the region for the sample. You need to add a buffer, and you can see there is a control line here, and there is a line for the immunoglobulin G, and there is a line for immunoglobulin M. This actually the slide used for the test, and you can see the different results. So basically, if the result came like this, there is no IgG and IgM, so this means that this patient or this individual have no antibodies, so probably it's negative for the corona. It's negative, so either it's not been infected. So the invalid result, you can see that all of these 4 different variety, it means that these are invalid results. So why is invalid result? Because if you need to understand the result, the control line should be present, because actually that means that this slide actually is working fine. In cases there is IgG positive, you can see only the IgG band will appear, as well for IgM. In case you can see double of these antibodies appear, so you can understand now how the different positive tests can be related to the figure. This is the region, of course, that you can detect both antibodies. By the end of week 6 and so on, you can only detect immunoglobulin G. You can early detect immunoglobulin M if it's only immunoglobulin M that is appeared. So actually, these are the references that I used for these lectures. Majorly, the major reference or the major textbook will be the BRS Microbiology and Immunology, and as well there are 3 recommended books for you to use, and these are the 3 recommended books. So, and this will conclude our 7th topic in Multisystem 1.